So in this work, we studied amyotrophic lateral sclerosis, that is the most common motor neuron disease, and it is devastating because so far there are no cures.
We focus on glial cells in ALS. First, in TDP43 mutant mice, we observed that gliosis begins early at the onset of the symptoms. By analyzing the RNA-seq of single glial cell populations, we observed a tendency to dedifferentiate, proliferate at the onset of the disease.
Astrocytes show an increased proliferation, an increase in GFAP levels and Ki67 levels, and this activation is transient, meaning that this is a transition from a proliferative state to a pro-inflammatory state with expression of pro-inflammatory genes. Importantly, we saw that this process is cell-autonomous. In fact, by deleting TDP43 specifically in astrocytes, we saw a reduction in gliosis and also a delay in motor symptoms and cognitive decline.
Answer on the mechanism that could drive astrocytes and oligodendrocytes to differentiate at the onset of the disease, we decided to exploit the GWAS data on ALS. And we came up with a prediction of hyperactivation for transcription factors bound to c-MYC.
We then tested whether MYC was truly hyperactivated in ALS astrocytes. While mRNA total levels and protein levels were unchanged, we then tested for the phosphorylation state of MYC, and we saw that this was dramatically changed. In fact, phosphorylation at serine 62 and threonine 58 is linked to an increase in activation of MYC transcriptional activity. Overexpressing MYC in astrocyte of mutant mice was sufficient to mimic the TDP43 pathology. This was observed in primary astrocytes from mutant mice, in impairs-derived astrocytes, and even in ALS-derived motor neurons. Conversely, inhibiting MYC with homo-MYC, a dominant negative peptide, was sufficient to reduce MYC transcriptional program and reduce gliosis. But how MYC causes neurons to die? We found that MYC is able to alter extracellular vesicle release from astrocytes that are not able to provide support to motor neurons. In fact, under MYC hyperactivation, AVs from astrocytes lose their trophic function and instead contributes to neuronal vulnerability. In summary, our works uncovers MYC-driven mechanism of gliosis that disrupt neuron-to-glia communication. This links human genetics to molecular pathology, demonstrating causality in vivo, and also identifies extracellular vesicles as both a mediator of neurodegeneration and also candidate as biomarkers. Most importantly, it highlights how MYC is a potential therapeutic target to reprogram glial cells and also to restore their supportive role in ALS.